Broadcast is now starting. All attendees are in listen-only mode.
Hello and welcome to today's BioPhorum webinar, an industry perspective on common practice in forced degradation studies of biopharmaceuticals. I'd like to start by thanking Nettie for setting up and running this webinar so efficiently for us. Although it would be lovely to hear from all of the audience, there are well over 100 of you now, so you've been muted to maintain good audio quality. If you'd like to ask a question, please do so by typing it into the chat box. You'll find that on your control panel during the presentation and indicate who the question should be answered by. The panel of authors and presenters will try and answer all the questions at the end of the presentation in a Q&A session. This call is being recorded and will be made available on the BioPhorum website at some point after the call. I'd now like to invite the other presenters to turn their cameras on so I can introduce them to you. So I'd like to welcome Surinder from BMS, Nick from Takeda, Goran from Boehringer Ingelheim, Christoph from Lonza, John from GSK, Lisa from AbbVie, and Jenny, our lead author from UCB. And my name is Elaine Stokes. Next slide, please, Nettie. We'll start the webinar with an introduction to BioPhorum and explain how this work came about. We'll cover the purpose of the work and address each stress style individually. We'll then pull together general and common themes and move on to additional considerations for non-mAb biologics. We'll look at how different stress types are used in the different stages of development and round up with conclusions before finally finishing with our Q&A session. Next slide, please, Nettie. BioPhorum has 8 forums where the global biopharmaceutical industry can collaborate to accelerate their rate of progress for the benefit of all. The development group is one of these 8 forums. Next slide, please. There are 13 work streams in the development group, and the development group's mission is to focus on better and faster process development for biopharmaceutical medicines. We successfully connect companies and provide a space where subject matter experts can work together on common targets. Of these 13 work streams, the forced degradation studies work stream is one, and I have the very great pleasure of facilitating this work stream that drafted the benchmarking surveys and discussed the output from those responses that ultimately delivered our common practice publication. Next slide, please. 5 years ago, the development group forced degradation work stream surveyed 12 collaborating member companies, and we had discussions around the responses and approaches to the design of biopharmaceutical forced degradation studies. The contributions come from companies of very different sizes and diverse product portfolios, and we believe that these results are a representative summary of overall industry practice. We aim to highlight the similarities and differences from these that came from the 175 questions that we drafted on forced degradation studies. This paper has now been published in the peer-reviewed journal of Pharmaceutical Science, and it was a very great honour for us to be selected for publication in the special edition that was dedicated to Professors Carpenter and Randolph. Next slide, please. I'd like to thank the authors that drafted this publication, and particularly those that are here today to present this webinar and be part of the panel. I'd like to acknowledge key members past and present who completed the surveys and contributed to all of the discussions that led to the article. Next slide, please. Forced degradation studies help to identify degradation pathways and main degradation products, and this supports critical stages in development, and it's used at all stages in product development. And you can see towards the bottom left of the slide, we aim to cover physical instability, And then just above that, you'll spot chemical instability, and we'll be covering that as well. The benchmarking survey highlighted the purpose of performing forced degradation studies: degradation mechanism understanding, stability assay development, assessment of critical quality attributes, formulation development, but also establishing comparability. And I'd now like to introduce Surinder, who will be covering thermal stress. Surinder, over to you.
Thank you, Elaine. And hello, everyone. My name is Surinder Singh, and I am a senior scientist at Bristol Myers Squibb. Today, in this webinar, I will speak to thermal stress in forced degradation study panel. As Elaine described earlier, forced degradation studies, directly or indirectly, are an essential component of overall control strategy that ensures safety, quality, and efficacy of the product. Thermal degradation is just one among various stresses in a panel of forced degradation studies. Thermal degradation has a unique capability to produce some of the most common degradants in one go, like particles, size variants, charge variant, and post-translational modifications. As you can see in an industry-wide survey results on this slide, the temperature used for thermal degradation studies varies from 25 degrees Celsius to 55 degrees Celsius. And the main driver behind this wide range is differential thermal stability of your protein or drug product and the goal of the study. In addition to temperature, there are other variables for those parameters needs to be considered wisely. Can we go to the next slide, please, Nettie? Thank you, Nettie. As you can see in an industry-wide survey results on this slide, there are other variables for these parameters needs to be considered. For example, the duration of the thermal stress. It can vary from 1 week to 3 months. Sometimes it can go even for 6 months as well. And then accordingly, number of time points will vary too. Then the container closure type and its position in the thermal chamber, the stress chamber, can vary too. You can have glass vials or you can have a Celsius pack depending upon the type of the material and their position. If it's glass vial, they can be lying horizontal or upside down. Then the type of material used for these studies can also vary. It can be some non-GMP material, or it can be GMP drug substance or drug product. Number of batches used in these studies also varies. It can be just 1, 2, or more than 2. And sometimes some other additional environmental factors needs to be considered, like humidity. Sometimes it is controlled. If it is controlled, its relative percentage can also vary. So the choice of all these parameters is primarily driven by the goal of the study. And the goal, for example, are we doing it for stability indicating method development or CQA assessment for comparability study, or are we doing it to support our shelf life claims? Whatever be the reason, the samples from different time points are pulled and subjected to a battery of analytical methods. And the information and insight gained from that analysis plays a central role in crafting an effective control strategy that ensures that the product is safe, efficacious, and is of high quality. So with that, I will turn it over to our next speaker, Nick, who will speak to another stress in forced degradation study panel. So Nick, please, it's all yours.
Thank you, Surinder. Hi, everyone. I'm Nick, and I'm the head of product attribute sciences at Takeda. Today, I'll be covering shaking stress and freeze-thaw degradation studies. When it comes to shaking stress, these studies are conducted by the vast majority of the companies to assess protein physical stability at air-liquid interfaces to support formulation, process development, or product handling. As seen on the graph here, nearly all of the companies surveyed reported conducting shaking stress studies using orbital shakers, whereas several respondents used different approaches to mechanical stressing, such as reciprocal, rotation, and shaking up and down. Other common themes include performing the shaking studies under ambient temperature, often with a control sample without agitation, and directly in the final drug product container closure and fill volume. For drug substance, it is important to use appropriate surface area to volume ratio to approximate its actual configuration. More variation is observed on the variables that include shaking speed, duration, and light exposure during the studies, which I'll highlight in the following slide. Next slide, please. Here's a summary of the shaking stress study design from the survey. Highlighting the more variable parameters, generally speaking, shaking stress is recommended at 200 to 300 rotations per minute for 24 to 48 hours. For proteins with different sensitivities to mechanical stress, the shaking studies could be extended or shortened to produce meaningful levels of degradation. A majority of the studies is performed under normal light exposure with 12 hours on and 12 hours off cycle. Certainly, if light sensitivity is unknown, it is advisable to err on the side of caution and protect the samples from light. The major expected degraded pathway is aggregation and particle formation, and correspondingly, SEC and subvisible particle characterization are the key analytical methodology of choice. Next slide, please. Freeze-thaw stress studies are also conducted by the vast majority of the companies, predominantly to support long-term product storage and shipping conditions, as well as to gain an understanding of the protein degradation mechanism and to develop stability indicating assays. Nearly all of the companies surveyed reported conducting the freeze-thaw studies in the upright position and undergoing 5 freeze-thaw cycles as shown in the pie chart here. More variation is observed in the freeze and thaw temperatures, presumably due to specific demands of the development project. In the selection of the freezing temperature, the product's glass transition temperature is generally taken into consideration, with most companies adopted -60 C or below, whereas -20 C is adopted to a lesser extent, as presented on the graph here. Next slide, please. Thank you. For the thawing cycle of the study, room temperature is the highly favored elevated temperature condition to maximize the effect on product quality over 5C. This slide is a summary of the freeze-thaw study design from the survey that also captures parameters that are equally split in practice, such as the fill volume, where it's either at the intended final drug substance or drug product fill volume, or at a scaled-down model, and the freezing speed control, where most companies do not control the speed, while others may control the freezing time or rate. The major expected degraded pathway here is aggregation and fragmentation, and correspondingly SEC and subvisible particle characterization are also the main analytical methods employed. Next, Goran will speak to pH stress.
Thank you very much, Nick. Hello, everyone. My name is Goran, and I'm head of laboratory in the Analytical Development Biologicals Department at Boehringer Ingelheim's Biberach site in southern Germany. Within this webinar, I would like to share the study results for pH stress and additional deamidation analysis. As for our force degradation studies as well, also a pH stress study may be conducted for multiple reasons and to answer multiple questions. For example, proteins may be exposed to different and partly extreme local pH ranges during production. As these conditions cannot be avoided and may occur at various durations, knowledge about their potential impact on critical quality attributes is essential. Furthermore, most biopharmaceutical products comprise liquid drug product formulations. Again, the pH value chosen for the formulation impacts product stability. Forced pH conditions may allow to quickly identify potential long-term degradation pathways. Finally, pH stress studies may, for example, also be conducted to just learn about the protein's liabilities in order to improve the critical quality assessment and to develop stability indicating assays. As outlined on this slide, the vast majority of companies apply basic and also acidic conditions in their pH stress studies. These conditions, on the one hand, depend on the physical chemical properties of the molecule, and on the other hand, on the purpose of the study. For basic pH conditions, a pH above 8 is common, and for acidic conditions, pH is mostly being set between 3 and 5. If feasible, pH is adjusted by titration. In case the desired pH set point is not adjustable by titration, also buffer exchange is being applied. The pH conditions chosen may be combined with the application of elevated temperature. Next slide, please. Thank you. Besides the conditions just mentioned, also a couple of other common approaches were determined by the presented BioPhorum study. You can find an overview of applied parameters in this table. Mostly, samples are being positioned in an upright manner and stress is being applied for 1 to 2 weeks. This incubation time is mostly covered by 3 to 5 sampling time points. However, incubation time may differ considerably from the most frequently used setup mentioned before, putatively due to the pH value and also the temperature condition used. Humidity is usually not controlled during a pH stress study, and studies are either conducted using drug substance or drug product in their respective container closure or in other unspecified containers. With respect to the degradations or modifications that may occur upon pH stress, the induction of deamidation, isomerization, and succinimidation is well expected on amino acid level. Furthermore, changes in higher order structure and the induction of fragments, aggregates, or particles are likely. These modifications determine the set of assays being mostly used for pH stress sample analysis. The modifications on amino acid level are monitored using CE and LC-based charge heterogeneity assays, but also mass spectrometry. The changes on higher order structure and the induction of fragments, aggregates, or particles are addressed by LC and CE-based size heterogeneity assays and also classical drug product particle analysis methods. The stress study data set is often completed using binding or bioassays to assess potential loss of function. Next slide, please. Thank you. In case common pH stress studies do not allow to elucidate especially deamidation sites and rates, additional studies to specifically address this issue might be useful. This could, for example, include very high or low pH values or the combination with multiple other stress types. To conclude, pH stress studies are another important component of the entire force degradation study setup applied during product development. Now, Christoph will take over and elaborate on details on photostress and chemical oxidation stress.
Yes, thank you a lot, Göran, for the transition, and also hello everyone from my end. I'm Christoph Garpentin. I'm a principal scientist in biologics formulation development at Lonza Drug Product Services in Basel, Switzerland. So during the development and manufacturing, during storage and handling, proteins can be exposed to light, which can then lead to protein degradation, for example, fragmentation or aggregation. The ICH guideline Q1b recommends stress conditions which contain a part for UV light and white or visible light. The test conditions provided by ICH were intentionally recommended for use with small molecules, but from our survey, it appears that most companies follow the recommendations also for protein photostress. For UV light, most companies apply the recommended 200 watt-hours per square meter, and some test even higher stress. For visible white light, most companies confirmed the use of 1.2 million lux hours as per the recommendation. As an additional outcome, the survey confirmed that most companies investigate photostress at ambient temperature or 25 degrees Celsius, with the humidity not being controlled. Studies are performed on both drug substance and drug products, and materials are kept in the intended drug substance or product storage containers in upright position. Next slide, please. We see here a fine breakdown of the survey results. Besides the exact stress conditions, which we described on the previous slide, it's interesting to look at the degradation observed and analytical techniques used. This is in the bottom 3 rows. Here you see that aggregation and oxidation are observed by almost all companies. Also, changes in functional assays are observed by 2/3 of the participants. For the analytical techniques, SEC, appearance, and particle analysis are applied. Interestingly, also 3/4 of respondents indicate to use ICIF or ICE to monitor photostress degradation. CSDS is used by 2/3 of the respondents. Almost all companies include a control which is not exposed to light stress. Next slide, please. On the next 2 slides, we want to look into chemical oxidation stress. Several amino acid residues are susceptible to oxidative degradation by various pathways, but no guideline exists on protein oxidation stress conditions. In our survey, we found that the majority of companies used forced oxidation studies to explore degradation mechanisms or pathways and also to support the development of stability assays. The expected degradation to be observed are naturally oxidation, aggregation, loss in functionality, or higher order changes. What most companies have in common is that they apply a buffer exchange or add a quenching agent in order to stop the oxidation reaction. But a huge variability and stress conditions is observed across the industry, probably depending on the pathway of protein oxidation that is of interest to be investigated. On the bottom right, we have assembled a few results for the duration and conditions applied to illustrate the significant variability in stress conditions. Next slide, please. We split the results further, and thus we can identify that hydrogen peroxide is the most commonly applied stress agent and is used in a very broad range of concentrations. Tertiary hydrogen peroxide is less commonly used than hydrogen peroxide. For metal-based oxidation, which in general is less applied, iron, copper, or mangan are being named. The duration of study is also very broad, ranging from a few hours to several weeks. The temperature applied is mostly 25 degrees Celsius, but also lower and higher temperatures are used. The studies are performed in the same container closure as the drug substance drug product or in vials or tubes. This may be dependent on the timing of the study, whether it's being performed early or later in the project timeline. In summary, we can say that protein forced oxidation is a stress condition found to be executed very heterogeneously across the industry with few common themes, and each company probably has their own recipe for stress, triggering the degradation pathway of interest. And with this, I would like to hand over to John to introduce us into results for general and common themes.
So thank you, Christoph. Hi, everyone. My name is John Campbell. I'm a scientific leader and fellow at GlaxoSmithKline in the United States. So I'm now going to discuss some of the general and common themes that arose from the results of our survey. So one of the things that we learned quite clearly was that multiple iterations of forced degradation studies are performed during the course of drug development. So this means that forced degradation studies are not done once and done, but they're repeated several times with the study designs being adapted to the information needed at that time of development. So these needs might include a number of things such as formulation development, development of stability indicating analytical methods, and identification of critical quality attributes. Now, there was a consensus on the anticipated degradation events that and the stress types which induce them. So I don't have sufficient time to go through all the information that we learned from this in detail, but on the chart shown here, what we have are specific types of degradation that might occur and the forced degradation conditions that are known to induce them. So you can see that aggregation is easily the most common route of degradation and can be induced by a number of conditions. Next most common was fragmentation, followed by oxidation, and then others. Next slide, please. So when a forced degradation condition was performed, each stress type was actually interrogated in a somewhat similar manner amongst the survey respondents. So for example, 3 to 5 time points are prepared typically for each stress type. Now, the duration of stress varied. It spanned from 3 months to less than a week and largely depended on the stress condition. So thermal stress tended to have the longest duration, while oxidative stress testing was the shortest. In terms of analytical testing of forced degradation samples, size exclusion chromatography and particles testing was quite common. And this calls back to the previous slide where aggregation was the most commonly expected route of degradation. So after these was capillary electrophoresis for monitoring size variants, including fragmentation, and then charge variant characterization techniques. So the analytical testing did vary depending on the stress type being interrogated and the particular goals of the study. Next slide, please. If you are interested in a summary of recommended force degradation study conditions, so that information is going to be shown on this slide. We have stress conditions, temperatures, durations, time points, containers, the need for controls, and other specifics for each condition. So much of this information was covered by my colleagues, but it's summarized here on this slide. Now, recall that this information pertains to monoclonal antibodies. And so one question we might ask and would like to now briefly address is how this approach might change for non-mAb biologics. Next slide, please. So it's difficult to prescribe, precise stress conditions a priori, but some general advice was gleaned from our survey, and it's kind of summarized here for proteins like Fabs, enzymes, antibody drug conjugates, and others. So for thermal stress, survey's respondents felt it was important to understand each protein's unfolding temperatures using, for example, differential scanning calorimetry. So ideally, survey respondents felt that stress testing should be conducted at a temperature in which the protein is not thermally unfolded, well below the transition midpoint, or TM. And so because we can expect TMs for most proteins to be lower than those of monoclonal antibodies, this will necessitate lowering the stressing temperature most of the time, we think. So no additional considerations were noted for photo or oxidative stress, but in general, it was felt that for physical stress, that is shaking and freeze thaw, less stress would be required. And then finally, for antibody drug conjugates, the chemical stability of the linker and the small molecule conjugate will play a role in selecting force degradation conditions. Next slide, please. So here, much of what I've just discussed is addressed on this slide, but with a few additional details, perhaps the most important being the need to consider additional analytical testing techniques required for non-mAb biologics. Next slide, please. So at this point, I want to come back to what I mentioned at the beginning of my section, which is that our survey showed that the designs of force degradation studies change as a drug progresses through development. So again, in the interest of time, I'm going to be brief on this slide, but for example, you'll see here that thermal stress is performed at all phases of development, while shaking and freeze thaw were reserved for mid and late phase development. And then likewise, photostressing and oxidation were emphasized in late phase as well. Next slide, please. Now, while it is clear that force degradation is performed repeatedly throughout development, most companies responded that they do not perform force degradation at every stage of development. So presumably, force degradation study is performed for specific purposes, the most common of which are listed here, including understanding degradation pathways, developing stability indicating analytical methods, evaluating critical quality attributes, formulation development, and supporting manufacturing comparability. Other purposes were reported, but these were less common. Now, because the point in development when these questions need to be answered varies, the stress types required to address the purposes might be different, and we think this is likely driving the results of the survey that I've discussed. It is just a matter of which question needs to be answered at which particular stage of development, and designing a force degradation study to answer that particular question. So in summary, I do want to apologize briefly for going through these slides fairly rapidly, but if you are interested in taking a closer look, I would encourage you to have a look at the paper, which contains this information, as well as some additional details that you might find helpful. And in addition, I also want to note that we intend to make this presentation available online if you would like to review the content at your own pace. So with that, I'll thank you for your attention to this point, and I will now turn the presentation over to Lisa, who will conclude it for today.
Hi, everyone. I'm Lisa Zhou from AbbVie. I'm a principal research scientist. I lead the new modality group in the protein analytics department. So here I'm going to bring you to discussion and conclusion. So the outcome of this BioPhorum development group force degradation point share survey on force degradation study is the first time data has been generated and gathered across industry to provide a greater understanding and a consensus approach for force degradation. Now, there is a lack of clear regulatory guidance on how and when and why to understand the force degradation study, only that they should be done to understand your product, but not very clearly how and when and why to undertake this force degradation study. Now, in the past, force degradation study data were used in a more selective and supportive role with the dossier. With the ICH Q8, 9, and 11, it has a subtle shift to the emphasis of our product-related understanding. Now that giving a greater role for force degradation study. More emphasis on CQAs and their control strategy through the whole process. And force degradation aid our understanding of the mechanism of degradation of our products, their susceptibility, their sensitivity, and crucially, the projection of samples to determine if our chosen assays are stability indicating. Also, more comprehensive product understanding is required. Next slide, please. So how to perform force degradation study? The details described in this paper highlights the options available to us to perform these crucial force degradation studies. These force degradation studies will always be somewhat tailored towards each organization, driven by their original selection criteria for development candidate. Now, in this paper, we have presented the consensus of the comprehensive survey summarized in the tabular format, which can be used by experienced or novice industry members alike to respectively compare to their force degradation practice or to inform and guide first-time studies on the logistic aspect of a force degradation study. Next slide, please. As for when and the why part of the force degradation can be utilized in all stages of the product life cycle. It can de-risk candidate selection and to generate samples suitable for determining the stability indicating nature of the release assay. It can inform formulation studies. It can help demonstrate the process comparabilities. It can develop understanding of the product degradation pathway. It also helps determine CQAs and even used post-approval under certain conditions. So overall, force degradation is really a very important part of the CMC product lifecycle toolbox to use as and when required for the product as deemed necessary. Next slide, please. So in conclusion, force degradation studies are an integral part of the product development lifecycle. Force degradation can be used from the selection of the molecule to lifecycle management. The data generated by force degradation study support a wide variety of activities during the product lifecycle. In this paper, we summarize the common practice used for force degradation study by the 12 corresponding companies now involved in the development of therapeutic antibodies, and biotherapeutic products. We also touch upon a tailored approach to alternative biotherapeutics. In final conclusion, we present this common practice as a general guideline for planning, setting up, and executing forced degradation at various stages during the biotherapeutic life cycle. So with that, I would like to pass it on to Ellen for acknowledgement. Thank you.
Elaine, I guess you're on mute.
Thank you very much. Rookie error. I'd like to thank the authors that drafted the publication, and particularly those who contributed to all of the discussions and those that are here today and have done such a lovely job of presenting the webinar. And they will be our panel for our Q&A session. So while our panelists are turning on their webcams, if you have any questions, please put them into the chat box. We're very, very keen to answer them and to have a discussion on the use of forced degradation in biopharmaceuticals. Next slide, please, Nettie. I'd just like to point out that the answers to these questions at the meeting represent personal views and are not necessarily the views of the other authors and their institutions listed on the presentation. So if you'd like to ask some questions, we'd be very happy to answer them.